So a couple nice things is that all of the FRQs that are used in the United States, every year they post them, and like this is off the internet, like you guys can get it. The scoring guidelines are off the internet, and then how students answer, like we've used in class before, that's on the internet too. So what I would do is I'd start with 2018 and work my way backwards, because the test is it's gotten much more molecular in the last few years compared to when it was like 2013, 2014. It feels there's a lot more DNA and gene regulation in it now compared to how it used to be. But looking at this. So a couple things here, they tell us like that there's a green allele that's dominant to purple, and then plant height is dwarf compared to tall. And then here's some of the important things here. We have individuals of the P generation, and P generation is always true breeding, and true breeding means that they are homozygous. But they gave us the genotypes of the parents of the P generation. But they produced F1 offspring. So, what would the genotype be of the F1 offspring? Yeah, so it's a dihybrid, meaning that the F1 would be heterozygote for both of those genes. So, they want you to construct a diagram below to depict the 4 possible normal products of meiosis that would be produced by the F1 progeny. So not by the parents, but by this genotype right here. We need to show the chromosomes and the alleles that they carry, so that means that you need to write the alleles on the chromosomes or indicate them in some way. And then assume that the genes located on different chromosomes, meaning that they're not linked, and that the gene for flower color is on chromosome 1. So that's important too. They're on different chromosomes, the gene for flower color is on chromosome 1. So here's chromosome 1. If we have an F1, so that means that we have chromosome 1, means that you get 1 chromosome from dad, and that's why I put it in blue. And 1 from mom. So I'm putting that in pink. The gene for flower color is G, so this is chromosome 1. And it does not matter what I make this to be. I'm just going to make the maternal one here dominant, and this is still chromosome 1, and this is going to be recessive. So they are homologous chromosomes, meaning that they have the same genes, but they might have different alleles. They're not sister chromatids yet. We also have chromosome 2, which is a little shorter, and since we know that they're not linked genes, I know that the other gene for height is going to be on that chromosome. So I'm just going to keep the paternal one with the recessive, just to make my life easier, and the maternal 2 is going to be dominant. This gives me a 2n or 4 diploid number of chromosomes. 2 copies of chromosome 1, 2 copies of chromosome 2, my diploid is 4. This would be G1. What happens in S? Yeah. I'm going to copy all of the chromosomes, and now they're connected at the centromere, but this is still chromosome 1, but now I have sister chromatids. This is still chromosome 1. They are sisters. Chromosome 2. Now I have replication of my chromosome. It will go through and we'll go to G2. What happens in G2? Yeah, so we have like preparation for division, but it is mainly about making sure that we have enough organelles and nutrients and everything else before we divide. So now we are ready to go into our first division of meiosis, because remember there's meiosis 1 and there's meiosis 2. So knowing that this was S, here we have prophase 1. In prophase 1, we form tetrads. And what that means is that our homologous chromosomes line up together. So here's my chromosome number 1. Here's homologous chromosome number 1. Together they form a tetrad. Blues are sisters. Blue compared to pink are homologs. What happens in prophase 1 that will shuffle my alleles? Crossing over. Another word for crossing over is synapsis. So, what that means is that genetic information can be switched between the internal non-sister chromatids. It's not going to happen on the ones outside, because if you're actually switching actual like matter, they have to be touching one another. So after prophase 1 and after crossing over occurs, I will get something that looks more like this. For part, I have my ones on the outside that stay the same, but my ones in the middle switch sides or crossed over. So initially we had a recessive G. I'm going to say that my recessive G moved over here, it crossed over. Here we had dominant G. Here's my dominant G that crossed over. And actually, like, I should probably, to make things like look a little bit better, like the loci or the location of the genes would like all be at the same spot. We're going to see the same thing occur on our bottom chromosomes, and we're going to have crossing over occur there as well. So far, are we good? So this is showing like the end of prophase, but I'm really also kind of showing what it would look like in metaphase 1, because here we have crossing over, which is one way that we have new genetic variation, but here we have independent assortment. Independent assortment means that I don't have all the paternal chromosomes on one side and all the maternals on the other. I have a mix. It's not like kindergarten when they said all the boys line up on the right, all the girls line up on the left. Like, it's just like everybody line up in 2 lines. We don't care how you assort yourself. Assort how you want. And that's what the chromosomes do. They assort randomly. Sometimes paternal are on one side, sometimes maternal are on the other. So when we go through our first division after anaphase, we will get 2 new cells. These are still sisters. Sisters stay together. Sisters before misters. But the homologous chromosomes stay together, separate. So what would look like? And what you should see is that each of these cells has like some that look like maybe the parent, but then they have this like recombinant one too. And then on this side, here's my G. Okay. So this is like after anaphase. Anaphase is when anaphase 1 when the homologous ones separate. Okay, so you guys with me so far? All right. So we need to do one more division. So we'll go through the whole process again. We'll go through prophase 2, metaphase 2, anaphase 2, and then afterward we will have 4 cells. And that's what they want you to draw. So the 4 cells that I have are just coming randomly from these. So I'm going to say that I have a lowercase g on chromosome 1. I have a dominant D on chromosome 2. So things that they're going to look for on the rubric. Everything needs to have a chromosome 1. So everything needs to have a G. But you should have 2 that have a recessive G and 2 that have a dominant G. How you place that, it does not matter. So there's chromosome 1 in each of those cells. Chromosome 2 in each of the cells should have Ds. But some will have a dominant D, some will have a recessive D. How they assort into these cells can be different. Like I don't have any cell here that has both dominant, right? But someone else might, and that's okay. As long as you have chromosome 1 and chromosome 2 in each, and I have 2 dominant Ds, 2 recessive Ds, 2 dominant Gs, 2 recessive Gs, how they shuffle does not matter. So here's the rubric. Each cell has 1 unduplicated chromosome 1. Each one has an unduplicated chromosome 2. Genotype combinations are there. As long as you have them, that's fine. The rest of the FRQ. Predict the possible phenotypes and their ratios in the offspring of a test cross between an F1 individual and it's a test cross, so we know it's homozygous recessive. So